In this video, we're going to look at membrane structure in assembly. So the way to think of the plasma membrane is that it is a fluid mosaic. That is, your membrane is constantly moving and it's more or less randomly distributed. It's like a 2D lipid sea. So your lipids are moving, your integral proteins are moving. So imagine that you're on a sea and your proteins are like the icebergs. Now, the reason we know that the fluid mosaic model of this random and moving lipid sea is real is through experiments such as this. So you have a mouse cell and you have a human cell. The mouse cell is covered in lipids that are fluorescing green, human cell fluorescing red. You fuse the two cells together, and immediately after fusion, half the cell is green, half the cell is red, as seen here in this photo. Let this cell rest for 40 minutes at 37°C, and then what you'll see is that the lipids will be randomly distributed. So here are the actual two photos of that. First, you test for green fluorescence, and you see it's all over the place. Then you test for red fluorescence, and again, you see it's all over the place. This shows that your lipids are constantly moving, and they are moving in a seemingly random fashion. Now, one way we can measure the speed of movement is through FRAP, or fluorescence recovery after photobleaching. This is how you can determine diffusion rates. And so what you do is that you attach a fluorophore or something that fluoresces to anything you want to measure in the membrane. Usually you use a mobilized cell or artificial membrane so you can actually measure this. Then over a small area of your membrane you shoot a laser. This laser, with its intensity, will destroy any fluorophore. That is, you no longer get fluorescence from the area that was hit. Then you see how long it takes until color recovers. That color is coming from adjacent lipids and/or proteins. And what you see with that, get my JavaScript out of here. What you see with that is that proteins travel at different rates. Roughly 30 to 90% of proteins are freely mobile, but there are 10% of proteins that seem just to be anchored to the lipid membrane. That is, they don't move, or they're extremely slow. And in the next videos, we're going to look at why some proteins are free to move and why some are tethered to the membrane. But I will see you in the next video to discuss that.